So that was just some brief introduction to PK and tox. If I can ask all the speakers to come back up to the stage, we're now going to have a Q&A session, so hopefully all the speakers are still here. And there's a seat for everyone. And we'll start the Q&A once people are up. So there's going to be some microphones coming around. So does anyone want to be bold and ask the first question? Kevin.
Thank you. Thank you very much to all the speakers. So this is a question primarily to Rachel and to Matt from the perspective of the new technologies. Just at a high level, what do you think? ISIS has been active since 1989 or whatever it was. Richard, I don't know if you... and ISIS has been active since 2000 and whatever. So they have a lot of experience in their respective areas. What can CRISPR and the messenger RNA approaches learn in terms of what went before? Because a massive body of data. What can they learn from that at high level, and basically what is maybe not can't be extrapolated? It's just too different.
I'll start, sure. And I mean, I think I alluded to this in my presentation, but I think delivery is not starting from ground zero. So from a real tangible perspective, these technologies, these new technologies get a boost from lessons learned in certain technological advances and things like delivery. I guess philosophically, you could also look at other things, and whether it's tractable paths to the clinic and the type of things that you might go after. I think as an industry, we learn from the paths of others. One notable thing is, it seemed like, it's my perspective, but there was a time when everybody took new technologies directly into cancers and trying to shrink tumors, humans, and I think people have evolved into more of the rare disease space and some of the other things that may be more tractable proof of concept. And we've certainly gone the path of going to vaccines as a kind of a lower hurdle into the clinic and starting to assess the technology there. I think gene editing, there's a lot of ex vivo potential applications and things like that. So philosophically, I think we just learn from what others have done and what works and what doesn't. So.
Yeah, I can only echo the delivery lesson, and I think not only will many of the companies use some of the platforms and technologies that have been developed for in vivo delivery, I think many will also say, we recognize how challenging that is, let's start ex vivo, where there are maybe more straightforward methodologies like electroporation to get into the cells directly without packaging. I also think, as actually you and I were briefly chatting about earlier, there's a lot of benefit in terms of manufacturing, right? If you think about the length of a CRISPR RNA, a tracer RNA, or a single guide RNA, yes, they're longer than the RNAs that the RNAi world needs, but it's building on top of that. And so I think there's a huge amount of commercial expertise for manufacturing that will be incredibly valuable for our field.
So my name is Edward Smith from Karolinska Institute. So I have a question. I think it was mentioned by Richard Geary about that there is a very slow elimination once these synthetic oligonucleotides go into cells. So what is actually known about this slow elimination?
Yeah, thanks. Good. So slow elimination systemically and in the brain is pretty much the same as nuclease metabolism. So for the modified oligonucleotides, which are generally phosphorothioate throughout and 2' modified on the ends, or throughout in the case of Spinraza, for example, the molecules are very slowly degraded by primarily endonucleases. Now, once the endonuclease cleavage occurs, you have very short or small oligonucleotides because it occurs in the gap, right? And so you end up with 10 mers, 9 mers, 8 mers, which have very low protein binding. And so they're generally some modulation or equilibrium between what's in the cell and what's in plasma. These small, short oligonucleotides almost immediately cleared through renal filtration and ultimately eliminated via excretion in the urine. So if you follow the oligonucleotide over time, do mass balance, you'll eventually accrue about 80 to 90% of an administered dose in the urine, but it takes months. And it's all coming out in the form of these smaller truncated oligonucleotides that have been endonucleolytically cleaved. And that's the basic mechanism for clearance.
Perhaps I'll just ask a quick question while other people are thinking of questions, or people are going with the microphones. I was quite surprised, Martin, it's coming to you actually, that you said 40% of your siRNAs are liver toxic. I mean, can you just expand on that a bit more? Why do you think such a high percentage are liver toxic? And other people on the panel may have some comments on that as well. I didn't realize that because I was thinking about screening. You know, if 4 out of 10 are liver toxic, how many people actually do that screening in the audience for RNAi-like molecules? So that was the background to it.
Thanks for the question, David. And I don't want to take too much actually away from the poster presentation we have tonight, but what we have discovered is that those liver toxic molecules, first of all, it's a very sequence-dependent effect. So the 60% which are not liver toxic show, based on their sequence, are very clean. On the liver toxic ones, I think there are grades, obviously, of toxicity. I showed you a fairly extreme example. But just in short, the sequence-dependent effects, we believe, based on our preclinical studies, are based on RNAi-mediated effects, basically off-target silencing of many other genes, but basically working like a microRNA.
Right.
And so it is a seed-mediated, we believe a seed-mediated effect, which causes, that's why it's sequence-dependent, and it can cause these off-target silencing and therefore the toxicity. I mean, of course, there could be other potential reasons, and we're looking into all of those. Do we perturb the microRNA pathway as a whole? That doesn't seem to be the case. That would also not explain the sequence dependence, because a lot of the siRNAs don't show that effect. And if you had just simply replacing microRNAs out of the pathway or the risk pathway, would mean that you would have a platform-wide effect with a potent siRNA. I don't know if that answered your question.
No, it's good to know. Anyone else want to comment on the panel?
Just to comment that also just with the oligonucleotides you have quite high attrition rate, which is certainly something that continuously improves because we just can also build the predictive algorithm in our design tools, and we see it also going down. But yeah, so I hope I could have advertised in vitro assays before you do in vivo studies.
I'll make one other pitch here for my colleague down the way. Actually, in silico, understanding these sequence-dependent effects can also help with defining or designing in silico techniques, which can screen out a tremendous number of these compounds.
Okay, another question. Right, yeah.
My name is Brian Johnston from Somagenics. I have a question for anybody that would like to address it, which is we've heard about how to avoid immune stimulation, and we've heard about how to build it in. And I'd like to know what the prospects are for combining these two, which would be useful, for example, for cancer or viral targeting.
Well, I'm happy to start on that, and then others can comment too. I think, you know, I was talking about this with the people from RigOnTech, for example, because they're trying to have a selective RIG-I activator, and my question was, why don't you allow TLR7/8 activation as well? And the challenge is that the different immune pathways may have different dose-response effects. And let me use an RNAi example rather than a RIG-I example. With RNAi or with antisense, you're typically dosing your compounds at doses in the mg per kg range. Immune stimulatory RNAs and DNAs are typically dosed at a few mgs. And so if you have immune stimulation in RNAi against hepatitis B virus, let's say, and you want to take advantage of TLR activation as well to get better activity against the virus. It's a little bit of a dangerous thing because you're going to get the immune stimulation at doses way below where you're going to be seeing the RNAi activity. And so calibrating that immune stimulation is tricky, and you may wind up with something that doesn't have a therapeutic index. That's probably the single biggest challenge, actually, in that. That said, I think that there is the potential to take advantage of multiple mechanisms, and especially, as we're getting compounds that are more and more potent with the GalNAcs, for example, and so forth, so that you can go down to very low doses. This may be something to come back to and to think about in any given program, because there is the potential for immune stimulation to be beneficial in some programs. I don't know if any of the others.
Yeah, I think when I think about our vaccine approaches, I think it actually takes advantage of that exact concept, right? Both in terms of infectious disease and the cancer vaccines. So in other words, if you took a messenger RNA vaccine that was really just crudely immunostimulatory, you're going to shut down antigen expression, right? You're going to actually have a PKR-induced reduction of antigen expression, which is not a good thing from the standpoint of vaccine development. So I think what we get right, maybe somewhat serendipitously, is this notion of immune stimulation in a way that is specific, that you still maximize antigen expression, but you trigger certain immune responses that are specific. I'm going to talk a little bit more about that in my presentation on Tuesday, but these chemical modifications that we apply to the messenger RNA, in certain cases, are not completely devoid of some immune stimulation. In some cases, we can get there for therapeutic applications, but for vaccines, that's probably not a good thing. But it's not the same as taking in just broadly immunostimulatory agents that shut down antigen expression.
See another question there?
My name is Hassan from Wolfson Childhood Cancer Research Center in Newcastle University. My question about the last talk from Roche about the co-culture experiment to filter out the toxic. Well, what happened to the feeder layer or to the niche? From my background and the research we are doing, we know that primary cells for specific type of leukemias, when we grow them in co-culture layer, and this is the only way to maintain them alive and cycling, they respond to the oligonucleotide therapy, which the project I am doing, and they respond differently from when we treat them with normal small molecule inhibitors. So will you be able to filter or to map what's happening to the co-culture layer? Because tumor microenvironment, especially in leukemia and in bone marrow niche, is very important for the development of the leukemia or to the cancer or any specific disease.
We just did some preliminary analysis in just characterizing the system a bit better, and we simply looked in the NPCs because a hallmark of them is like activation of Kupffer cells, looking into cytokine secretion in order to just see if they are or if the functionality is still sort of intact, and that was the case. So we could see induction of cytokine secretion with the cells and all that, but otherwise we didn't look a lot into other factors or didn't play around also with for example, changing the ratio of the cells. And so I think there is a lot to play around with.
So it was a simple co-culture of one
It was just a simple co-culture. Yeah, it was no
Okay.
additional niche matrix effect. So it was.
Okay. Thank you.
Other questions? Ryan?
Hi, my name is Ryan Senton. I'm a graduate student in Dr. John Rossi's lab at City of Hope. I had a question for Dr. Hart or anybody in the in silico prediction of ASOs. So current RNA folding algorithms don't typically take folding as coming out of the ribosome, where you would imagine, the thermodynamic forces are greatest. They kind of just predict it after the fact. And additionally, they don't tend to take RNA binding proteins into account. So then that leads to the question, how much of the target RNA is actually exposed or thermodynamically unstable enough to be targeted by an ASO? And then would taking the time to perform a more laborious protocol like RNA shape be an alternative for a therapeutic company when you're trying to design ASOs?
That's a great question. In terms of structural predictions, in terms of the folding and how well that predicts ASO activity, in model systems we've seen it predict very well, but when you try to extrapolate it out to new targets, it tends not to always predict well, and I think it's partly because the way the oligos work, we know we get activities both in exons and introns. So if you think about if we're getting activities in introns, that means that we occasionally at least have strong activity in a co-transcriptional process. And co-transcriptional, as you alluded to, co-transcriptional structure is something that's very difficult to predict, and even using shape-seek techniques to capture the RNA as it's coming off in a co-transcriptional process is pretty challenging because the introns just don't last that long. So it's a real vexing problem actually to understand what the structural biology landscape of a transcript looks like and how that corresponds to ASO accessibility. That said, as I showed, when we look across the transcript map, depending on the target, we can get activity in a variety of places. So it doesn't seem to correspond to like all exons or all introns or 3 or 5' UTRs. It's really a target-to-target variation.
Other questions?
Hello, my name is Mehdi Qabomi. I'm from Copenhagen University. And I had a question regarding to CRISPR-Cas9. As we all know, CRISPR-Cas9 is a really specific nuclease, and it can, as you showed in your presentation, it's actually, but it's not a specific in addition because the repair system adds some nucleotides in the break site. My question was that have you considered to check whether the repair system prefers to add, for example, A next to the G when we break the DNA in G site, if maybe repair system prefers to add something like A or T to the G or prefers to add T to C site?
Yes, good question. So we have looked at that. It depends. If certain nucleotides are present immediately proximal to the break site, it significantly biases a given nucleotide being inserted. In other cases, it's almost completely random. So it's clear that the DNA repair machinery engages with certain nucleotides in a very specific way that leads to a certain type of insertion in some of the sites.
Any further questions from the audience?
Maybe a follow. I'm Vineshwarya with Om Life Tech. A follow-up for the ASO people. So I think we mentioned in case of gapmers when they are, cleaved. Do you see a reason when they're cleaved? Sometimes, we have this modification at the ends. Do you think that once we have gone from 20 mers to 5 mers, do they act as steric blockers sometimes because now, do non-specific binding and all those things?
Give it a shot. So the 5 mers, once generated, appear in the urine fairly quickly. So I'm not sure exactly what the question is, whether they can be active or whether they can...
Yes, and how long are they before they're excreted out? I mean, whatever time they're in the cells, do you think they can cause some...
Yeah. So when we take snapshots at, say, 24, 48, 72 hours, we actually don't find 5 mers in the cells. So we presume, because we're seeing them appear in the urine and we don't measure them in tissue, that they're fairly rapidly excreted once formed. So we're not catching them in the cells. But even if they were in the cells, and we've done certainly in vitro and cell culture work with smaller and smaller oligonucleotides. Once you get down to a 5-mer, you're not going to get activity, certainly not at the doses and the small percentages of those doses and concentrations that the 5-mers exist.
Any final questions? We've got sort of 5 minutes left. If anyone has a burning question. If not, we can finish. I'm sure the speakers will, if you want to come and ask one of the speakers a question, why don't you just come up to the front and have a quick chat. I'm sure they're willing to stay for 5 or 10 minutes and just answer any specific questions. So if anyone has got a burning question that they want to ask but didn't want to ask it in the general forum, just come up to the front and speak and talk to the speakers. But I'll close the session here then, and thank you all very much, speakers. Fantastic session. You all kept to time, and it was a very tight agenda, so very well done, and some great talks. And just to say, I think the presentation has been videoed, and the presentation will be going on the website, so that's one of the reasons you may not have been able to note down all the references, but I did ask the speakers to include references in their talk, so hopefully you'll be able to see the slides on the website in due course, and therefore you'll be able to pick up the references from there. So hopefully you've all found it a useful and educational session, and we look forward to seeing you now in the main meeting, which will start at 2 o'clock. So thank you, everyone. Thank you again, speakers.